And welcome to our 40th Great Lakes Phragmites Collaborative webinar, part of our Students of Phragmites webinar series. I'm Ned Willig from the Great Lakes Commission, and I'm hosting the webinar today on behalf of the Great Lakes Phragmites Collaborative. Our presenter today is Courtney Robichaux from the University of Waterloo, who will be discussing measuring invertebrates and aerial insectivore birds after Phragmites australis control in a freshwater marsh. Before we get to our featured presentation, there are a few housekeeping items I'd like to review, and just talking a little bit about the Great Lakes Phragmites Collaborative. So the Great Lakes Phragmites Collaborative, for those who don't know, is a partnership to link people, information, and action on invasive Phragmites. The collaborative is coordinated jointly by the Great Lakes Commission and the USGS Great Lakes Science Center, with funding from the Great Lakes Restoration Initiative. The collaborative facilitates communication amongst stakeholders across the region, and we assist in the development of management tools and programs and serve as a resource center for information on Phragmites biology, management, and research. The collaborative is led by a multi-agency advisory committee and applies principles of collective impact to advance a common agenda for Phragmites management in the Great Lakes region. Another way the collaborative engages with Phragmites professionals is through the Phragmites Adaptive Management Framework, or PAMF. PAMF utilizes participatory science to provide data-driven Phragmites management guidance through an adaptive management framework. What this means is that we have established a way to learn from Phragmites managers who are doing what Phragmites managers are doing and turn that data into annual guidance to help managers make effective and efficient management decisions. We encourage anyone who's currently managing Phragmites in the Great Lakes Basin to consider joining PAMF. We are planning to host a live virtual training session for PAMF on May 25th from 10 to noon Eastern to help interested managers get involved. Registration information is available on our website, but please reach out to us at pamf@glc.org if you would like additional details or would like to hear more about the program. In addition to our virtual training session that will be live, we'll be hosting a self-guided training resource through the Moodle learning platform that will be available at the beginning of April. The Great Lakes Phragmites Collaborative recently released its Common Agenda. This Common Agenda is the first step we are taking to align our regional goals and establish a shared vision for change. The purpose of the Common Agenda is to clearly define the intentions of the collaborative, to develop a shared understanding of gaps and problem areas surrounding Phragmites, and to highlight the areas and strategies that we are working on or wish to work on in the future. We recently hosted a public briefing that discusses the Common Agenda, as well as provided an update on recent collaborative activities and outlined the steps that we would like the Great Lakes Phragmites community to take in order to help us advance our Common Agenda. A recording of that webinar is available on our website, and again, that's www.greatlakesphragmites.net. There are several steps, though, that you can take to help us make the progress on our Common Agenda. First, we are asking Phragmites managers in the region to participate in PAMF. After completing the training for PAMF, participants with a management unit managing Phragmites typically spend about 3 to 5 hours annually participating in PAMF, but this small time investment really helps have a large impact on our collective learning effort and helps managers across the region. I know a lot of you joining us today have many years of managing Phragmites or research experience with Phragmites, and if you have resources that may be helpful to other Phragmites professionals, please consider sending them to us through our listserv or sending them to us at phragmites@glc.org for us to host on our website. Considering how widespread of an issue Phragmites is, both geographically and in terms of impacts, learning and sharing information on management Phragmites is going to give all of us the best chance of success with Phragmites. We encourage you to coordinate management efforts with others in your area, and finally, to consider how your activities and the activities of your agency could be aligned with the collaborative. We'd really like to hear from you on your work and how that can contribute to advancing the Common Agenda and what we can do to help facilitate that as well. you're interested in learning more about Phragmites management and research, you can check out the resources on our website, which include management guidance and recordings of past webinars. You can also join our listserv to connect with hundreds of other professionals and sign up for our bi-weekly newsletter, which includes a quarterly research roundup of academic papers published in previous months. You can find links to join these on the top of our website, which is www.greatlakesphragmites.net. Today's webinar will be recorded and uploaded to our YouTube channel and website. You can access an archive of past presentations on our website and also view and register upcoming webinar presentations using the URL listed here. So hello to those of you who are watching the recording. Please share with your colleagues and don't hesitate to reach out if you are watching this long after the presentation date. And of course, connect with us on social media. Tag us on Facebook or Twitter at GLC-Frag or GL-Frag as the Twitter handle. To help our webinar run smoothly today, I will now go over just a few technical reminders. Audio for this webinar can be streamed through your computer speakers or telephones. And to use a phone line, find the call information in the meeting connection materials that were sent out prior to the webinar. In order to minimize potential interruptions and background noise, we have automatically muted all attendants. Please keep yourself on mute and your cameras turned off to save bandwidth. If you're experiencing technical difficulties, please send a message to my colleague, Sam Tank, at phragmites@glc.org. We ask that questions be submitted any time during the webinar using the chat feature. Our speaker will answer all questions at the end of the webinar. We will do our best to take all questions or comments, but if you need to leave the webinar before we have gotten to your question, we encourage you to review the webinar recording that will be posted on our website. So finally, it's my pleasure to introduce our presenter. Courtney Robichaux is a PhD candidate at the University of Waterloo, where she's been studying wetlands invaded by Phragmites australis since 2015. She's especially interested in how we can better understand the risks to Canada's freshwater systems and ensure that they are protected. After her PhD, she will be joining the Orell lab at Queen's University to study how microplastics affect freshwater food webs. Thank you so much, Courtney, for sharing your work with us today. I will transfer presentation over to you now, and please take it away.
Thank you so much for that introduction, Ned. And do you mind just giving me a thumbs up if that looks good? Can you see the slides?
Looks
awesome. Great. Okay, well, thank you all so much for joining me here today. I'm going to be sharing with you a little bit of work that I've been doing since 2017 in particular, looking at how macroinvertebrate communities differ between invaded marsh, herbicide-treated sites, and remnant uninvaded marsh, and if that relates to aerial insectivore bird use within these marshes. And this is my PhD work, but I couldn't have done it without the help of my research technician, Jacob Basso, who's currently at the University of Guelph, and of course, my supervisor, Dr. Rebecca Rooney. So just before I get started, I want to acknowledge everyone who helped make this possible, including the research assistants, the lovely partners I've had the opportunity to work with, and of course our funding agencies. I also want to take a moment to just acknowledge the land that I work on and that I currently live on. So Longpoint is the traditional land of the Mississauga of the Credit First Nations, and Waterloo and the University of Waterloo, both where I live and where I work, is situated on the Haldimand Tract. And this was land that was promised to the Haudenosaunee of the Six Nations of the Grand River. And I wanted to draw people's attention specifically to a sort of conflict going on the Haldimand Tract, which is since July, Haudenosaunee land defenders have been peacefully reclaiming land to stop a housing development just south of Caledonia. And if you have the time at the end of this, I'd love it if you could look up, they've got a Facebook page, a Twitter page, and you can help contribute to their financial needs or the needs of the camp. And this is really the forefront of conservation that's so important. They're protecting our land directly from development. And on that note, so many of you are probably aware that we've lost a lot of wetlands globally. Wetland loss is rampant, and it's also a problem that's present in our own backyard. So in southern Ontario, we've lost over 80% of wetlands, typically due to development, agriculture, and other human activities. And wetlands are biodiversity hotspots. They do a lot of other important functions for us in the landscape, but I'm going to be focusing today a little bit more on the biodiversity that they support. So wetlands are incredibly productive, and they provide habitat for specially adapted wetland species, as well as lots of other species that use wetlands for even just a small portion of their life cycle. And in Ontario, Southern Ontario, where my work takes place, we have some particularly beautiful and diverse coastal wetlands around Lake Erie. So my work occurs in the Longpoint Peninsula. This 32-ish kilometer sand spit is located on the north shore of Lake Erie, and it actually represents about 70% of the remaining intact coastal wetlands on this north shore on the Canadian side of Lake Erie. And as such, it's got a lot of protections. It's a provincially significant wetland. It's a World Biosphere Reserve. It's an important bird area. All of these protections prevent development in the area or prevent any alterations to these important habitats. However, laws and policies and legislation make it a little bit easier to protect these wetlands from development, but it's more challenging to protect them from degradation that can happen because of an invasive species. And in Longpoint in particular, there's a large issue with Phrygmites australis. Now, I assume to this group it needs no real introduction, so I'm just going to give you a little bit of background about the invasion of Phragmites into Longpoint. So this perennial invasive wetland grass began to spread exponentially in Longpoint around the late 1990s, likely facilitated by low water levels that exposed substrate that really allowed it to establish and begin to grow quite rapidly. So there was some genetic testing. It was likely the haplotype M that was introduced and began to rapidly spread in this area. And it's now well established in the region. So I think in 2015, 2016, some aerial imagery from Ducks Unlimited estimated that in most or in some portions of the marsh and where I am working in the kind of western portion of the Longpoint Peninsula, Phragmites covers about 70% of the marsh. So it's really taken over and displaced our native vegetation communities. And this, of course, has well-documented negative effects on wetland biota. And I'm showing you two pieces of research here that were conducted in Longpoint specifically. So this work by Chantel Markle and Pat Chow-Fraser found that endangered blandings turtles actually used, Phragmites in the home range of endangered blandings turtles actually reduced the size of their home range, and that nesting females didn't always necessarily avoid Phragmites, but they would often try to still kind of power through these stands, and the researchers often found them desiccated or stuck in those stands, and then there was higher mortality, or there's the potential for mortality. And this is some work that myself and my supervisor, Rebecca, did following up on a study from the early stages of invasion, where we assessed the wetland bird communities using invaded marsh and uninvaded marsh, and found that species of conservation concern, like this Virginia rail here, so wetland obligate species that are in decline around the Great Lakes, we rarely ever found them in Phragmites habitat. So while Phragmites is not necessarily an ecological desert, it's not supporting the sort of, I suppose, higher quality community composition of wetland birds we come to expect from these beautiful intact coastal wetlands. This, of course, presents a problem when we're trying to sort of preserve these coastal wetlands. So in 2016, the Ministry of Natural Resources and Forestry, the Nature Conservancy of Canada, and other partners began this rather ambitious project to start to control Phragmites in both Longpoint and in Rondeau Provincial Park, which is another sand spit a little bit south on the coast. And I'm just going to be talking about Longpoint, where in 2016, with an aerial application using helicopters, I believe they controlled about 400 hectares of very dense Phragmites using a custom glyphosate-based herbicide. Now, this is a herbicide with a custom surfactant, so it should be safe to apply over water. But this is the first time in Canada that we've applied a glyphosate-based herbicide to control Phragmites australis directly over standing water, and we're doing this in a biosphere reserve. So we really want to understand how biological communities respond to this, or how they're using this herbicide-treated habitat. And as we're probably familiar with, wetlands are really productive ecosystems, but they're also deeply interconnected. And what I'm going to be talking to you about today are the macroinvertebrate communities, many of which have an aquatic life stage, and then they will emerge into flying adults. And the aerial insectivores, the swallows that rely on them as prey. Now, of course, there are other species that rely on these macroinvertebrates, and they play an important role in the food web, and they're not only prey. I feel I have to say that, but I will be talking about them in the context of prey items today. And for those of you who are a little less familiar with macroinvertebrates present in aquatic ecosystems, I thought I'd put together this little show of the life stages of a dragonfly, a macroinvertebrate we're probably all fairly familiar with. So invertebrates can be decomposers, they can be predators, they could be prey items. The dragonfly in particular is predaceous. So they have a good portion of their life as these aquatic nymphs where they are swimming around, they're predators. In wetlands where there are no fish, they can often be the top predators in that food web. And then they will, at a certain point, emerge out of their sort of they'll metamorphosize, they'll emerge, they'll leave behind this exuviate, and then they'll have a life stage as a flying adult. And this is really what I'm interested in here for my research, capturing both the aquatic invertebrates, those who live all of their life in the aquatic ecosystem, so let's say snails or worms, and also those that have an aquatic life stage and a flying emerging life stage. And these are in particular important food items for aerial insectivore birds like swallows. And swallows do preferentially use wetlands as foraging habitat. And aerial insectivore birds, such as swallows, have seen some of the sharpest declines across North America. So since the 1980s, they've seen about a 60% decline in their populations. And this is often attributed to multiple factors, but the decline in invertebrate prey items is often thought of as a contributing factor. And this leaves kind of a large gap of when wetlands are invaded by Phragmites, how does that alter the macroinvertebrate communities and the potential prey items for these species? And when we treat Phragmites, when we cause this rather large disturbance by removing a lot of living biomass in the hopes that a native community will reassemble and take its place, how does that affect the macroinvertebrate communities? And there's a lot of, we really want to know what is kind of happening here so we can make decisions around what's best for the native biota when we're controlling Phragmites. So my research that I'm presenting to you today has kind of three components that I'm going to share with you. The first is, what does the invertebrate community look like in Phragmites australis? What does it look like in the herbicide-treated sites? And how does this compare to uninvaded remnant marsh? The second is, how do aerial insectivore birds use Phragmites australis, use the herbicide-treated sites, and use uninvaded marsh? And the third piece is combining both of these, which is when we look at strictly only the flying invertebrates, the count of them, so the number of them or the amount of biomass in each habitat, does that relate to the aerial insectivore foraging activity? So can we kind of predict they're using this habitat more because there's more prey items? Share with you briefly the approach we took to answer these questions. So I set up 27 sites in Longpoint and sampled them in 2017 and 2018. And I've got a little map here. We're in the western portion of Longpoint. And I compared, so they were set up in Phragmites australis stands, in remnant uninvaded habitat, and in sites that were treated with this glyphosate-based herbicide in 2016. So the samples I'm showing you are 1 year and 2 years sort of post-herbicide treatment. And at each site, we collected invertebrates and also conducted point counts for aerial insectivores. And these were diurnal point counts. The radius for them was rather small. They were only, I think, 50-meter radius point counts. They were tight. We wanted to make sure that they were still sort of directly assessing how the birds were using the habitat. And what I'm showing you today is recording of birds that were only actively using the habitat. So we ranked them all on a scale of 1, 2, or 3. I'm showing you the birds that were deemed to be actively foraging in the habitat or that we saw successfully catch something in the habitat rather than simply flying straight through. And a little bit more in depth about our approach to macroinvertebrates. So in 2018, we wanted to specifically capture the aquatic invertebrates, so that potentially larval stage or just aquatic invertebrates that live their whole life underwater. So in May 2018, before large emergence events started, we went out and did a Quadrat column core sampling procedure. And that is, at each site, we collected a soil core, 2 water column cores, and we also collected all of the submerged vegetation within a 1/4 meter squared quadrat to capture macroinvertebrates that were using these different microhabitats. And then for the emerging invertebrates, we set up these tents, these emergence traps in May, and we left those up until the end of July. And we went out and collected the collection bottles every 10 days, and that would be the same day that we did our point counts. So we have a continuous measure of macroinvertebrate biomass at these sites, emerging biomass at these sites, measured with, or sorry, paired with sort of these timestamps of aerial insectivore point counts. And then to assess the prey quality and quantity for aerial insectivores, we looked at only the taxa that actually had an adult flying stage. So we took out all the spiders, for example. We counted all of them and we weighed them so we can get an idea of the number of prey and the amount of biomass for prey. So for the first question, what does the invertebrate community in Phragmites look like compared to the herbicide-treated sites compared to the remnant marsh? So what I'm going to show you first is a comparison of Phragmites australis invaded habitat in the purple and uninvaded remnant marsh, which is the yellow sites here. And what we found was there is actually no statistically significant difference in the abundance, richness, or evenness. So that's sites weren't particularly dominated by one taxa. Each taxa was sort of evenly represented. So there wasn't much of a difference in abundance, richness, or evenness between Phragmites australis and remnant marsh. And this was true for aquatic invertebrates on the top row here, as well as emerging invertebrates on the bottom row. You might notice, for example, for emerging invertebrates, there's slightly higher abundances in a few of the remnant sites, but again, none of these differences were statistically significantly different. However, when we include the herbicide-treated habitat in this analysis, you can see that for both aquatic invertebrates and emerging invertebrates, there was significantly higher abundances in the herbicide-treated sites. And this is combining both 2017 and 2018. So this was a trend that was present in both years, significantly higher abundance in the herbicide-treated habitat. This abundance, however, didn't translate over into the other measures of community composition. So for aquatic invertebrates in particular, the taxonomic richness was similar between all 3 habitat types, and the evenness was also similar between all 3 habitat types. So if we're looking at these metrics, there's not much of a difference between Phragmites habitat, remnant marsh, and herbicide-treated sites. However, when we looked at the emerging invertebrate taxa, which was, just to remind you, this was collected over both years for the whole field season, May to the end of July, you can see that taxonomic richness and evenness were both lower in herbicide-treated sites. So this means there was really high abundance of macroinvertebrates in the herbicide-treated sites, but that high abundance was represented by only like a few very dominant taxa. And one of those very dominant taxa was chironomids, or these non-biting midges. And what I've done here is kind of pulled out from the emergence traps over the field season the abundance of chironomids within the Phragmites habitat, the uninvaded habitat, and the herbicide-treated habitat. And you can see we caught that early seasonal pulse, that big pulse of chironomids, but they were fairly high in our samples. So I mentioned one of my research texts was like, you should definitely put this in, because in all of the samples that we looked at, there was about 20,000 chironomids in the herbicide-treated sites out of about 30,000 chironomids total. So they were very, very abundant in the herbicide-treated sites. Now, of course, this is interesting, but the other thing that I want to show you is the community composition of all of these sites. So this here is a piece of an ordination for the aquatic invertebrate communities. And this is just representing each site as a point. Sites that are closer together have more similar community composition than those that are further apart. And then I've overlaid some taxa vectors, and these are taxa that were pretty strongly correlated with the habitat type. And what you'll notice is there were no taxa that were very strongly correlated with invaded Phragmites australis. For the herbicide-treated sites, we had snails, hydrozoans, so these are aquatic, like fully aquatic invertebrates, and also chironomids and mayflies were pretty strongly associated with herbicide-treated sites, as well as a longhorned caddisfly. So these chironomids, this is the aquatic larva of those non-biting midges that I showed you earlier. And these little mayflies down here were also associated with herbicide-treated sites. And then the remnant marsh had a little bit of a more, not necessarily on this one, so there's spiders, there's columbola, but there's also rove beetles and then some scavenger water beetles, so some semi-aquatic invertebrates and fully aquatic invertebrates. And there is fairly high diversity in those remnant marsh sites. And this was, again, the aquatic invertebrate samples that I showed you earlier. Here, however, is the emerging invertebrate communities, which is emerging invertebrate communities in Phragmites were very similar to those in remnant marsh. So there was no difference really in those community compositions. There's a lot of overlap, and likely this, I need to pull this apart a little bit more, but I think likely it's because there's canopy height and standing dead are associated with these higher diversity, which is there's a lot of emergent vegetation in both remnant marsh and in the Phragmites australis marsh. And macroinvertebrates in wetlands are typically pretty resilient. We'll note we only went down to a family taxonomic level, but they're pretty resilient to the natural variation. So likely these generalists or these more resilient macroinvertebrates, they can do fairly well, or they're associating with just sites that have a lot of emergent vegetation. But what I think is really interesting around the herbicide-treated sites is that we had chironomids that were very strongly associated with the herbicide-treated sites, and they were also associated with high water temperatures. Now, high water temperatures at our site meant that there was not a lot of emergent vegetation. So there's nothing to intercept the sunlight. These were typically sort of shallow open water sites that had a lot of maybe submersed vegetation, but there wasn't a lot of an emergent canopy, as opposed to some herbicide-treated sites that had a higher abundance of mayflies. These were actually associated with Chenoplectus pungens, which is 3-square resh. So these are some of the herbicide sites where there is a little bit more emergent vegetation. And then we see the macroinvertebrates that use that emergent vegetation to sort of climb up, shed their exuviate, and become the flying adults. Chironomids, on the other hand, they can kind of just float to the water surface and emerge there, so they wouldn't require emergent vegetation as much as the other taxa. So we can kind of see that interesting divide of the composition of the macroinvertebrate community in the herbicide-treated sites that don't yet have a lot of emergent vegetation is really dominated by these chironomids. So now I'm going to kind of change gears a little bit and compare aerial insectivore bird use among these three habitat types. And what we found was consistently aerial insectivore birds were foraging more often over the uninvaded marsh and the herbicide-treated sites compared to Phragmites australis sites. And we saw 6 species, commonly barn, tree, cliff, bank swallows, purple martins, and eastern kingbirds. Tree swallows were by far the most abundant. But we did find that they were foraging less often over the Phragmites australis habitat. And I've pulled this out for you all to sort of show trends over time in the periods where we collected them. So this is collection periods 1 through 6, and I've again combined the 2 years. But we have tree swallows here, which had the highest abundance, and you can see that they were almost consistently across the entire field season, foraging less often over Phragmites australis habitat. Similarly, barn swallows almost consistently across the entire field season, both years, they were foraging less often over Phragmites australis habitat. And so this is interesting. It doesn't necessarily add up with the community composition that I showed you. So we thought maybe looking at only strictly the flying emerging invertebrates, can that be directly related to the aerial insectivore counts? So this figure here is we've pulled out only the invertebrates that they're flying, so strictly flying invertebrates. And one is the count and one is the biomass. And you can see that again, the count of flying invertebrates is significantly higher at the herbicide-treated sites, likely again driven by those non-biting midges. For biomass, again, there's more biomass of emerging invertebrates at the herbicide-treated sites, but there's actually a little bit more biomass in the invaded sites of these emerging invertebrates compared to the herbicide-treated sites, which is interesting. And now when we relate the two, this is kind of the pattern or sort of non-pattern that you see. And the one on your left, that's emerging invertebrate biomass compared to the aerial insectivore bird count, so the total count. And the one on your right, that's the emerging invertebrate count, so just the number of prey items compared to the aerial insectivore bird count. And you can see for the herbicide-treated sites, there was higher biomass, there was higher invertebrate count, and there was a pretty consistently kind of higher amount of aerial insectivores counting there, or sorry, aerial insectivores foraging there, in particular compared to Phragmites australis habitat, which even the sites that had the slightly higher biomass, there's not necessarily a clear relationship between higher biomass or higher counts and more aerial insectivore birds. What really kind of throws it or is sort of not unexplainable, but kind of doesn't really show much of a pattern as the remnant marsh. So we can see that the biomass is fairly low and the invertebrate count is fairly low, but there's a wide range of aerial insectivores foraging at those sites. So the count range from 5 to 10 to nearly 100 birds. And this is likely because, or really what this is showing us is that likely prey is very important, but also habitat characteristics. So more invertebrate prey, higher counts, higher biomass are likely important, but also habitat characteristics are probably really important for these aerial insectivore birds when they're choosing where they want to forage or spend more time foraging. In particular, here's some photos of what the herbicide-treated sites where a lot of aerial insectivore birds were foraging, and these are, like I mentioned, kind of shallow open water marshes. They've got some emergent vegetation, but there's a lot more interspersion than in a really robust cattail marsh, or in particular, a dense stand of Phragmites australis. Similarly, the uninvaded sites that we measured, they did span a water depth, so they include both meadow marsh all the way to emergent marsh. And these sites have also a lot more interspersion, a lot more sort of heterogeneous canopy heights. So from low to high to little open water ponds, typically there were trees or shrubs sometimes nearby, particularly in the meadow sites that were useful for roosting for these birds. So it seems to be a combination of both prey quantity, prey quality, if we're looking at biomass as a proxy for that, as well as the habitat characteristics of these sites. So the kind of concluding takeaway messages are that for emerging invertebrates, the communities were actually very similar between Phragmites and uninvaded marsh, likely because, at least at the family level, these aquatic macroinvertebrates are fairly resilient generalists who can kind of accommodate whatever emergent vegetation there is present. After herbicide treatment, there's an incredibly high abundance of chironomids in those sites for at least 1 and 2 years afterwards. Likely because, again, these are fairly generalist taxa. They can handle disturbance and they can handle sites that don't have a lot of emerging vegetation. And the takeaway is that creating good foraging habitat for aerial insectivore birds, swallows in particular, likely is a combination of both the prey availability and the habitat characteristics. But our findings were that aerial insectivore birds, they were foraging more often over remnant, uninvaded marsh, and herbicide-treated marsh, rather than Phragmites australis. So that's kind of that's everything I have to share with you today. And it's kind of it's nice to see that there are endangered species are foraging more often after we have treated and removed Phragmites, even after just a few years of treatment. But it is something to keep in consideration that these large-scale disturbances of removal of Phragmites will have an impact on the macroinvertebrate community. You'll see a different aquatic invertebrate community, and you will see different emerging invertebrates, a slightly less diverse community. So it's important to think about when you're treating Frag to sort of not do it necessarily all at once, leave some refuge habitat for these macroinvertebrates. The nice news is that there doesn't seem to be any particularly unique value of Phragmites australis. So if the remnant marsh, if the sort of refuge habitat you're leaving is uninvaded habitat, then that should be enough of a refuge for the diverse macroinvertebrates that do require emergent vegetation, unlike chironomids. And on that note, I just thought I'd put up, this is some of the other work that I've published. Some of it is also related to this work looking at vegetation community response and glyphosate concentrations after herbicide treatment. And I have also my information here if any of you are interested in following up or looking at my website where I try to put up some plain language summaries of these things. But in conclusion, thank you all so much for joining me today, and I look forward to hearing your questions and comments.
Excellent. Thank you so much, Courtney. That was amazing. We now have some time for questions. So it looks like the first question we've gotten so far is, so were macroinvertebrate samples kept? Are there any plans to refine IDs to genus or species level?
They are absolutely kept. I don't know if there's any plans. That's honestly more of a Rebecca question as my supervisor. I think it would be a whole nother graduate degree for someone, like potentially a whole other PhD, but it would be really fascinating to go down to genus or species level and see if there are, like, I expect when we go down to that finer level of taxonomic resolution, we would probably see more differences than what we capture at the family level, but no immediate plans.
Great. Are there any plans for longitudinal studies on treated areas extending beyond a couple years?
Yeah, so the work that I'm doing, so just because of I'm in graduate school, I've only worked up till 2018, but our lab has actually been working really closely with the Ministry of Natural Resources and Forestry, and now with the Canadian Wildlife Service, where they're treating additional areas in Longpoint. And that is actually all more longitudinal work. So there's continued measurements of, in particular, the plant community responses to treatment. They're doing 5 years after treatment, and I'm not sure how long they're doing for the Canadian Wildlife Service parts, but there is plans to do longer than just 2 years afterwards. For macroinvertebrates, I don't think there's anything in place for long-term monitoring, just because they are so much more research intensive in terms of identifying them. It took us as much time in the field to collect them, and then it took as much time in the lab to identify them. But I agree, it would be really fascinating to watch as the vegetation community kind of recovers in the treated sites to see if the macroinvertebrate community also recovers and begins to resemble remnant marsh. That would be really fascinating. Yeah.
Awesome, thanks. Did you see any differences in response of birds of special interest, like listed species or conservation target species?
So for these field, so what I'm presenting here was we only did aerial insectivore bird surveys, we did them, they were diurnal, so we did them during the day because that's when these birds are foraging. So many of these species are listed, like the barn swallow is an endangered species, but we didn't observe, because we weren't doing the appropriate sort of methods to capture any other wetland bird communities. However, there was recent work published by Doug Tozer and others at Bird Studies Canada, where they have actually looked at the response of marsh birds to the treatment of Phragmites, which I could post in the chat at the end of this. And they've actually found marsh birds, including those of conservation concern, like I think it's green heron and others, have responded fairly positively to the control of Phragmites.
Great. So we have a couple questions related to the treated sites. And so were any of the treated sites a complete Frag monoculture before treatment, or had all had some interspersion?
So for this project, because they were, it was the aerial application of herbicide, they treated just dense monocultures of Phragmites. Now, in these marshes, because they've been, like Frag has been expanding since the late 1990s, there's like the leading edge of patches, but all the Frag that's present is fairly dense. And all the Frag that was treated in this study was very dense monocultures that could be treated with the aerial application.
Okay,
Yeah.
and in the treated areas, was treated Frag still standing dead, or had the biomass been removed?
Great question. So in Longpoint, in the treated areas, the biomass had been, the majority of it would be rolled or cut. So we had, yeah, most of it, just thinking back to our sites, most of it was rolled. So it wasn't standing dead anymore. There was some sort of secondary treatment. It was a little bit of a mosaic of what was rolled and what was cut, I think, depending on what machinery could get in there, but it was all in some way, the standing dead biomass was removed in some way or pushed down in some way.
Great. Are there any plans to extend this work to amphibian or reptile populations that also feed on macroinvertebrate communities?
Yeah, that's also a really great question. So no plans that I know of. I'm not sure if that might be incorporated in future monitoring for what perhaps the Canadian Wildlife Service is doing. The paper that I mentioned before, actually evaluating the response of birds, of wetland birds to this control, they actually also looked at amphibians. They looked at frogs in particular, it was an evaluation using the marsh monitoring program. So there has been a little bit of work, but I don't know if there's plans for direct work yet about the relationship between, let's say, prey availability and herptiles out there.
Okay. And related to that, has anyone looked at bat foraging responses to Phragmites treatments?
Yeah, I've gotten this question a couple times when talking about this work.
Mhm.
Not that I know of right there, but there is a lot of, or like a decent amount of bat work in Longpoint. And I think it would be a really like incredible idea for some collaboration there to look at how those other important aerial insectivores are using these habitats. Yeah, but I don't know of anything off the top of my head, sorry.
Yeah, sounds good. What impact do you think the Great Lakes water levels had on your results?
A pretty large impact, to be honest.
Mhm.
So the work that I've done has been, we've also been monitoring the vegetation community that's coming back after the treatment of Frag. And in a lot of, so I've been working out there since around 2014, and in a lot of the sites where Frag was treated, they were surrounded by meadow, like meadow marsh. Uh, Frag was treated in about 2016 when Lake Erie water levels have started. They've been kind of above historical averages since then. And a lot of those sites have really flooded. So what's come back has been a lot of submersed aquatic vegetation, emergent vegetation, which now they're shallow open water wetlands is probably selecting for all the flooding is selecting for a slightly different community of aquatic macroinvertebrates. So I think that's definitely has a large part to do with what we're seeing right after treatment. That is kind of a natural part of these coastal wetlands. There should be years where the water levels sort of draw down, we see that reemergence from the seed bank and we see more meadow marsh. But right now, yeah, they're very flooded. So it's like shallow open water wetlands, and that I would say absolutely has something to do with the macroinvertebrate community, which is another reason why kind of longer term monitoring would be really interesting to see both what the vegetation community does when water levels go down and how the macroinvertebrate community responds to that.
Great. I just want to note my colleague Sam Tank shared a paper about the control of invasive Phragmites increases marsh birds, but not frogs. So that's in the chat if people are interested in that. We have one more question here. Do you know if there are any plans to monitor fall roosting birds?
Again, I'm not sure if there's any plans in particular for monitoring the fall roosting birds. I know they do tend to roost a lot in the Phragmites stands, and the removal might reduce where or change where they're able to roost. There is still a lot of sort of typha. There's still a decent amount of patches of Phragmites that we're working on removing. Yeah, it would be really interesting to see because these dense Phragmites stands are fairly like on an evolutionary timeline. They're like a blip, right? They've only been here since the late 1990s. So it'd be really interesting to see if they're like the natural history records of what these birds were roosting in before Phragmites was present, how they're using Phragmites now that it's available, and then to see if sort of they still have habitat available to them once you remove Phragmites, like if there's still an abundance of the trees they were using previously or the emergent vegetation that they use. But yeah, it would be really interesting to monitor, keeping in mind that, yeah, Phragmites in these marshes it's only, what, 20 years old, late 90s is, oh God. Yeah, it's like, so it hasn't been around for their entire evolutionary history. So I imagine they would adapt fairly rapidly to its removal, but that's just conjecture, I suppose.
Great. So some aerial invertebrates display a diurnal versus nocturnal flight preferences. Did you take this into account regarding bird prey availability?
In terms of prey availability, no, that's actually a really interesting question. So we only did diurnal point counts, so it might be that we're underestimating the foraging activity of the aerial insectivore birds that do have a more nocturnal or even like a crepuscular sort of foraging preference. The thing about the emerging invertebrates was we measured those, so starting in May, essentially continuously around the clock until the end of July. So we have a measure of kind of the total amount of the number of invertebrates and the biomass of invertebrates. But we wouldn't be able to separate out like if there's different pulses in the day, which, yeah, so this work is really has been really fun to do, and we had a lot of questions that we're sort of trying to get answered. And I think like a lot of good science, it basically just means, okay, so we sort of answered one question and now we have 100 more questions to kind of poke in and pull apart all of these patterns that we're seeing.
Yeah, there's definitely a lot there. Are there any other questions that we have? Not seeing any. I just want to thank you again, Courtney, for answering all these questions so thoughtfully. This has been really, really amazing and great to learn some more about this. We will be uploading this webinar in the coming days, so we will send a follow-up email when the recording is available on our website. So for those that see this and want to share it with other people, feel free to refer them to there. We will also be having many more of these Students of Frag webinar series to come. The next one is April 28th at 1:30 p.m. Eastern, and students from Wilfrid Laurier University will be sharing about their work doing a spatial analysis of Phragmites in Lake Bernard in Ontario. So we hope that you will be joining us for that. I again want to acknowledge the USGS for their support in this through our grant through the Great Lakes Restoration Initiative. Again, this webinar will be posted on the collaborative website in the next few days. And if you have any other follow-up questions or comments, please feel free to contact us at phragmites@glc.org. So that concludes today's webinar. Thank you for joining and have a great day. Bye.
Thanks. Bye.